Hey everyone! In this video I am going to describe DNA replacement in Neurospora crassa. So if we want to replace a certain interval of DNA in the Neurospora crassa genome, we will have to do a genetic transformation. And when we do that, normally we first culture the fungus, Neurospora crassa, in a tube of medium like this or a flask containing media. Now, after several days, the fungus will grow and it will make millions of these asexual spores called conidia. So here I'm showing a single conidium, and the conidia in Neurospora crassa have between 2 and 5 nuclei. And so here I'm showing 1, 2, 3, 4, 5 nuclei in this hypothetical conidium. Now, Neurospora crassa is a haploid organism. So each nucleus is haploid, has a single set of chromosomes. So if we were to look in the nuclei, we should see 7 of these chromosomes in there, 7 Neurospora chromosomes in each nucleus. Now, if we were going to replace a particular interval of DNA in the genome, we could call the DNA that we wish to replace, we could call that the target locus. So here is a hypothetical target locus. And let's say these are the base pairs of the locus here, and it doesn't matter too much, but the A's are blue, the C's are orange, the T's are pink, and the G's are yellow. And here we can have the base pairs, and I'm showing the bases as these little squares, and the phosphodiester backbone as these lines here. And we can see the base pairs, and DNA is double-stranded, and we all know that by now. So for conceptual purposes, let's say we want to replace this region right here in the crassa, in the Neurospora crassa genome. So this is the target, all of these bases here. And in this example, that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. 16 base pairs. So we want to precisely replace these base pairs with a different sequence. Now, in order to do that, we will need to make a DNA replacement vector. We will need to construct a DNA replacement vector. And these go by multiple names. Sometimes they're called knockout vectors, sometimes they're called transformation vectors, but technically, I guess the most accurate term is a DNA replacement vector. Because what we're going to do is we're going to replace these base pairs with a different DNA. So when we construct these vectors, what we typically do is we look to use the flanking DNA on either side of the target locus. So let me show you that in this diagram here. So let's say we want to replace this sequence right here. Well, in order to do that, we would need 500 to 1,000 base pairs of DNA that matches the sequence to the left of the target. So we would call that the left flank. Now, in reality, the DNA replacement vector will use a left flank that is, let's say, 500 to 1,000 base pairs long. For this conceptual exercise here, I'm using these 1, 2, 3, 4, 5, 6, 7, 8 base pairs to represent 500 to 1,000 base pairs of sequence. And so the left flank is going to match this sequence exactly. It's going to be identical. It's going to be identical. So if we want to replace this region right here, and we want the point where the replacement starts to be to the right of this, well, we would need 500 to 1,000 base pairs of sequence to the left of this point. And that's what I've diagrammed down here. You can even match up the colors if you want. So I think we go to right here. Pink, blue, blue, pink. Okay. Here we go. So that's the left flank. Now we would need to do the same thing for the right, for the region to the right of the interval that we would like to replace. We would need 500 to 1,000 base pairs of sequence to the right of the interval. So that we would call that the right flank, and I have that right flank down here. So if we have 2 DNA molecules, one the left flank, one the right flank, each 500 to 1,000 base pairs long, and each either matching the sequence to the left of the target interval or the right of the target interval, we would almost be ready to replace this sequence, replace the target locus. The next step is to fuse the left flank and the right flank to what we call a center fragment. So let's see, I have this right here. So let's say this is the center fragment we're going to fuse this to. So there are many different ways. So in this case, we would have 3 DNA fragments, the left flank, the right flank, and what we're calling the center fragment. And there are many ways to fuse these. We typically use a PCR protocol to fuse them using sort of overlapping sequences, and I'll cover that process in another video. And I hope you can see this. I hope it's in focus. So the center fragment in this case right here, for this conceptual exercise is rather short, right? But in reality, it would be about 1,000 to 2,000 base pairs long, and it would encode an antibiotic resistance gene. Usually it encodes an antibiotic resistance gene. So after fusing these together, well, that would be our complete DNA replacement vector. Now the center fragment, and this is going to come into play, I think, in the next video where we try to predict the genotypes and the PCR products resulting from, well, I'll get to that in the next video. But point is, the center fragment does not need to be the exact same size as the target that we're trying to replace with the center fragment. It could be much longer, it could be much shorter. Usually it's not the same exact size. Okay, so I hope you understand that so far. Now, once we have our DNA replacement vector constructed, and we have enough of the DNA molecule to do the transformation, well, we can transform the fungus. So we would isolate the canidia, so probably millions and millions of canidia, could be up to 1 billion. And we wash the canidia with a special solution. And, okay, so we have our center fragment here. Well, no, I'm sorry. We have our DNA replacement vector here. And here I've diagrammed the DNA replacement vector as having the left flank and the right flank, with the left flank 500 to 1,000 base pairs long, right flank 500 to 1,000 base pairs long, and their center fragment usually 1,000 to 2,000 base pairs long. And that changes depending on the antibiotic resistance gene that you were using in the DNA replacement vector. So you'll mix these two together, or we mix these two together when we're transforming the fungus. And so you can have tens of millions of copies of the DNA vector and tens of millions of canidia in the solution. And so what we do is we put these in a little cuvette called an electroporation cuvette. We put that in a machine called an electroporator, and it shoots an electric current through there. Oh, one thing I wanted to point out is this is not to scale, right? So these DNA molecules, usually the full length of the DNA replacement vector, usually, let's say 3 kb to 4 kb long, right? And this is a nucleus. These little blue things, these are the nuclei. So obviously the DNA vector is not to scale relative to each of these canidia in this diagram. But my point is, you mix these together and you shoot an electric current into the solution, and somehow, miraculously, we still haven't worked out all the details of how this works yet. The DNA will go into the canidium, into the nucleus, and sometimes into the chromosomes. Okay, so we're using an electric current for that. Okay, so let's say the DNA gets into the canidium, gets into the nucleus. Maybe the nucleus was undergoing mitosis or something, the nuclear membrane broke down. We don't really know for sure how the DNA replacement vector gets into the nucleus, but let's assume it gets into the nucleus. And let's say of the 7 chromosomes, let's say this chromosome right here has the target locus that we are hoping our DNA replacement vector replaces. So here's the DNA molecule. Now, somehow, the DNA replacement locus is replaced because the left flank matches, right? The left flank of the target locus, and the right flank of the DNA replacement vector matches the right flank of the target locus. And through a process which likely involves DNA, what we call DNA recombination proteins. These proteins mediate the exchange of DNA. And they mediate the exchange of DNA because the left flank here matches this left flank here, matches this sequence here, and the right flank here matches this sequence here. And it probably, if you took BSc 350 with me, it probably has something to do with homologous recombination repair proteins. But the important point is, because DNA is exchanged here, this DNA replacement vector goes in and replaces the DNA at this location. As a result, we have the DNA replacement vector now integrated into the chromosome, and the DNA that it replaced, well, it ends up outside, not integrated into the chromosome, and most likely degraded by exonucleases or endonucleases, but either way, degraded. And so that's lost. Okay, so we have the DNA replacement vector, the antibiotic resistance gene, inserted into the genome. Now, so that happens at a very, very low frequency. We could say maybe 1 in 10,000 canidia. Maybe 1 in 10,000 canidia has that target DNA go to the desired location and replace the desired interval with the antibiotic resistance gene. So let's say these represent 10 million canidia, 10 million canidia. And let's say this one canidium right here represents the one canidium that was transformed. Transformed meaning that uptook the DNA and the DNA integrated into the target location and replaced the desired DNA at the target location with the antibiotic resistance gene. So there are multiple nuclei in here, right? Only one of the nuclei was transformed. Only one of the nuclei is what we will call the transformed nucleus. The others are not transformed. So we have to take those 10 million canidia and we have to spread them on selective medium. So the selective medium contains antibiotic. In our laboratory, we almost always use an antibiotic called hygromycin. And the antibiotic resistance gene we are using in our DNA replacement vectors confers resistance to the antibiotic hygromycin. And so if these represent 10 million canidia and only this one is the one transformed nucleus, all of those millions of canidia, they won't germinate, they won't grow because they're not resistant to the hygromycin. They don't have the hygromycin resistance gene. But this one right here with the transformed nucleus, well, it has that resistance gene, so it's able to grow. So it's able to grow on that medium. And so after a few days, what we'll do is we'll notice, hey, we've got some growth here. We've got some growth here. I should probably say that this is not to scale either. These canidia are so small you wouldn't be able to see these canidia with the naked eye. But once the canidium starts growing and forming a little colony, well, we'll see that. And what we'll do is we'll cut that out of the plate and we'll transfer that to a culture tube. We'll transfer that single, what we'll call that single transformant here, to a culture tube containing antibiotic again. And if it's a real transformant, it will keep growing. If it truly has the antibiotic resistance gene, it will keep growing on this medium. Now, we're going to call this the heterokaryotic transformant. Why? Because look here, it has the red transformed nucleus, but it also has some non-transformed nuclei. So those non-transformed nuclei still have the copy of the DNA we were trying to replace with the antibiotic resistance gene. So the heterokaryotic transformant has multiple nuclei. It has the non-transformed nuclei and the transformed nuclei. So what we need to do is isolate homokaryotic offspring. That's one of the easiest ways to isolate a homokaryotic, let's say, a transformant, is to isolate an offspring from the transformant. And those offspring are almost always homokaryotic. Let me explain how that works. So here is our heterokaryotic transformant growing in a test tube, making lots of the asexual spores. What we would need to do to isolate a homokaryotic offspring is to isolate the conidia, transfer them to a suspension of water, and then use little droplets of the water containing the conidia to fertilize what we're calling female protoparesthesia of a mating partner. So we grow the mating partner in a medium called crossing medium, and after 7 or 8 days, it forms the female protoparesthesia, which are the immature fruiting bodies, and we can fertilize these just by dropping droplets of water containing conidia from the heterokaryotic transformant onto the medium. Sometimes when you do crosses like this, the protoparesthesia producing parent is called the female strain, and the conidia producing parent is called the male strain. So this is the fertilization process. So we're fertilizing the protoparesthesia with the conidia. After about a month, that cross will produce what we call mature fruiting bodies called paresthesia. They will shoot their ascospores out of the paresthesia, out of the fruiting body, onto the lid of the crossing plate. Now, I said ascospores. So we already mentioned one spore type, conidia, right? Conidium is the singular of that. So these are asexually produced. So there's no sexual reproduction involved with forming these types of spores. The ascospores are the offspring of Neurospora crassa. They are produced by sexual reproduction between two parents. So these ascospores right here are shot to the lid. We can suck them off the lid into some water. We can make a water suspension of the ascospores. We can store these at 4 degrees Celsius pretty much indefinitely. They last a very long time, some decades. Some people think decades or up to 50 years. So they're very stable in water in the dark at 4 degrees Celsius. When we want them to germinate, what we need to do is heat them at 60 degrees Celsius for 30 minutes and then plate them on a medium that has hygromycin. Why do we want medium that has hygromycin? Because ideally, half of the offspring will have the non-transformed nucleus and the other half will have the transformed nucleus. Ideally. Because the male conidia were heterokaryotic, the number of offspring that have the transformed nucleus is actually going to be proportional to the number of transformed nuclei in the male parent. I probably just really confused you there with that one. But long story short, what we need to do is plate these ascospores on the medium with hygromycin and look for the ascospores that are growing well on the hygromycin medium. So here's one that's trying to grow and it's not having much success, so this is probably dying. And here's one that is growing very vigorously, so it probably has the antibiotic resistance gene and is a homokaryotic strain containing only transformed nuclei. So what we would do, we would pick this, we transfer it to a culture tube, and that's our homokaryotic offspring. Now, why is it homokaryotic? It's because, let's say this is an ascospore. This is an ascospore. So I already showed you previously what the conidia would look like if you had a pretty powerful microscope when you're able to look. The conidia look like this. They're sort of circular, they're kind of irregular circles, sort of. Well, the ascospores are these spindle-shaped or American football-shaped spores, and they are usually darkly pigmented. But each ascospore is formed from a single nucleus, right? A single nucleus that undergoes mitosis to make many nuclei. But because all the nuclei come from a single nucleus, all the nuclei have to be the same, So this is a neat trick we use to get homokaryotic transformants. Technically, they're homokaryotic offspring from heterokaryotic transformants. But once we have the homokaryotic offspring with the transformed nuclei, we can now do our tests to see how replacing that DNA changed the phenotype of the fungus.